Okay, we will go ahead and get started. I want to welcome everyone to the second lecture of our Spring 2021 Cofrin Logan Center Seminar Series. I want to welcome everyone who's part of our CLC family here at the University of Kansas, and also extend a special welcome to our friends of the center who are joining from other departments at Kansas, as well as other universities and organizations all over the United States and elsewhere. For those of you joining us for the first time, I wanted to share that the Cofrin Logan Center for Addiction Research and Treatment at the University of Kansas was established in 2018 as a hub to bring together researchers, practitioners, KU students, and community partners to address challenges in addiction through research, treatment, and community outreach. Before I introduce our speaker today, I want to explain a little bit about how this webinar will work. So Dr. Hugh Garavan will be presenting his slides on the screen, and he'll have a video and voice for his talk. A selection of attendees have the option of turning on their video feed, while other attendees will not have video. We ask that everyone keep your microphones muted unless you've been selected to ask a question at the end. At the end of the talk, we do expect to have some time for some Q&A. It will be moderated by myself and the other hosts. Go ahead and submit your questions in the chat window for the panelists to see and consider. If your question is selected by one of the hosts, we'll call out your name, we'll unmute you, and you can ask your question to Dr. Garavan. Unfortunately, we don't have time to get to all of our questions usually, but we try our best to get them. And sometimes we prioritize different orders. So if yours doesn't get asked, it might still get asked at a later time. I do want to thank Sergey and Drew for their coordination and logistical support for today's seminar and the rest of the series. And we'll also ask Sergey to put our Twitter feed and our CLC events page in the chat window for you so you can see the rest of our speakers coming up this spring. We guarantee you won't be disappointed for who's left this semester. And now I have the distinct pleasure to introduce today's speaker, Dr. Hugh Garavan, or as he says, call him Hugh, from the University of Vermont. Hugh is a professor of psychology and psychiatry at the University of Vermont and a member of the Vermont Center on Behavior and Health. I received a master's and PhD in psychology from Bowling Green State University. His research focuses on brain structure and function and how they relate to individual differences in psychological abilities, and has a long-standing interest in addictions as well. And he's especially interested in the changes that occur in brain function during the adolescent years. He's the co-developer of the Addiction Working Group for the Enigma Consortium, which for those of you that don't know, this is a big data initiative that brings together MRI data from over 100 labs in 30 countries. And he is one of the leaders of the Addiction Working Group for that consortium. And his most recent work is focused on being associate director of the massive multi-site adolescent brain and cognitive development study funded by NIH. And he's the co-PI of the University of Vermont site of that study. As far as addictions neuroscientists go, I think Dr. Garavan is in a truly rare class. He is somebody that I remember reading research of his when I was in grad school and postdoc. He's been consistently funded by NIH and other agencies. And what I think is really impressive is his papers have been cited almost 30,000 times, according to Google. And he has 4 papers that are pushing 1,000 or over 1,000 citations each. And so this is a really unique opportunity for our center to hear about cutting edge addiction neuroscience research from one of the world's true preeminent cognitive neuroscientists. So if I haven't embarrassed you enough, please join me in welcoming Hugh to our talk today. Thank you very much.
Yeah, thank you. Yes, you have embarrassed me. You've been Google stalking me. I should warn everybody, Michael promised me that he would filter out any difficult questions. So if you want your question to be included, make it the softest, easiest question possible. Okay, great to be here. Thank you. It's a shame I'm not there in person, but hopefully soon. While I was waiting to get started, I realized I have a very grandiose title here. So we'll see if I can deliver on causes and consequences of cannabis use. Okay, so hopefully, let's get going. There we go. Okay. I didn't realize that there's dictation at the bottom, so we'll see how well it does in capturing my my Irish accent. What I'm going to do is talk about cannabis use, and primarily in adolescence. Because of the audience, I'm going to give all of just one slide, trying to convey why this is important, because I know you're all very, very familiar. So, with regards to, say, focus on brain development, we know that there are lots of interesting brain changes and substantial brain changes that happen during the adolescent years. I'll come back to these data later. This is from the IMAGEN study in Europe that has been following kids for 10 years. We know that CB1 receptors increase dramatically during puberty. We know from animal models that adolescent tissue might be especially susceptible to any kind of damaging effects of cannabis or THC, more so than adult tissue. So there's a lot of reason to think that this might be an important period to look at. You all know this. Cannabis use continues to be, fairly common amongst adolescents. You also all know this. There's ongoing decriminalization and legalization of cannabis in the US, in Europe, elsewhere. You know that the ratio, cannabis is getting more potent. Ratio between, THC and cannabidiol seems to have increased, I think, like 60-fold in some examples over the last few years. And then finally, the adolescents' perception of the risk of cannabis has continued to dramatically decrease. So, we have a situation where it might be useful to investigate whether or not cannabis may in fact be damaging for the developing brain. And, that's what today's focus is. But, causes and consequences, this is challenging, right? I mean, we all know this. I'm not telling you anything you don't know. Human research, typically correlational, very hard to figure out. Even if you do, use a neuroimaging tool like functional MRI, are you seeing brain associations that are causes? Are they consequences? Or are they just, unrelated to the drug use itself? It's hard to determine. Causes and the consequences. There might be very, very many. They might be very varied. There might be different causes, different consequences for subgroups. Any one cause or consequence might be very weak when studied in isolation. The research you mentioned, the ABCD study, I mean, one of the things that's definitely coming through so far is that true effects tend to be very small effects. The most robust, reliable effects that we see, if you do proper cross-validation, are small. And very, very many studies have been underpowered. And, neuroimaging, I think, is reckoning with this right now, that our studies and therefore our findings have, let's just say, statistically underpowered heretofore. And then, of course, there's also the concern substance users and your comparison groups tend to differ on many, many confounding variables. A longitudinal study in and of itself is not going to, identify causes and consequences. It'll help. But, the whole point is just kind of get us to step back from the grandiosity of the title. I'm going to tell you what causes and consequences are, is just to convey this is challenging. This is going to be tough. And, I don't know what your sense of it is, but cannabis use really gets an awful lot of people hot under the collars. Some people are very much in favor of decriminalization. Some people think it's the devil's brew. So it's a contentious topic, and and getting guidance from the researchers is challenging for all these reasons. Let me give you an example, just to kind of make this a bit more concrete. So this is a set of analyses we've done on amygdala function cannabis. So again, this is in teenagers. This was published a number of years ago. What we did, this is from the IMAGEN sample. What we had, this is a paper published back in 2015. So one of the nice things of having a very large sample is, as well as finding, kids of interest, in this case, 70 cannabis users, you can then go and hunt for well-matched controls. So what we did is we identified 70 kids at age 14 who reported using cannabis. And then we found 70 controls who were matched on sex, handedness, age to the week, verbal comprehension, perceptual reasoning IQ, puberty levels, socioeconomic status, whether they had any alcohol, any cigarette use, and which site they came from, because this was a multi-site study. And the task here was just kind of a passive viewing. I don't know if you can see that here, but essentially it's a little video. There's an actor who does something kind of ambiguous. In this case, licks his lips or something, or, closes his eyes, as opposed to in another condition, emotes and, an expression, in this case, gets angry, and then we have a control condition. We compare the cannabis using kids and the controls and looked at the amygdala, and we see interesting differences. So just to unpack this, right amygdala, left amygdala, we have the angry videos, we have the neutral. You see controls don't show much difference. Cannabis users show this heightened response to the angry stimuli in the right amygdala, and also same effect over here in the left amygdala. Okay, so we have here, evidence of, neurobiological differences, functional brain differences between cannabis users and controls. Great, and you can come up with your own post hoc explanation as to what might be, what this might mean. The challenge, of course, is how do we know, was this effect caused by the cannabis use? So let's skip forward. These kids are part of this longitudinal study, so we have studied them again 5 years later. So what we did here is we identified kids who at age 14, when we got those brain images, who were all cannabis naive, but who then went on to use cannabis, so between the ages of 14 and 19. And we're not talking here about cannabis use disorder, but just, if you will, typical recreational use, if you will, by adolescents. So you'll see some are reporting 1 to 2 lifetime uses up to 40 plus. So again, all were naive at 14, but they went on to use. We then went and looked at their amygdala activity at age 14. And it's noisy, but there is a significant linear trend here. My mouse is bouncing all over the place. There is a significant linear trend here showing that in kids who were naive to cannabis, but who went on to use cannabis, their amygdala reactivity to those angry faces was predictive of future cannabis use. So again, it would suggest that that amygdala hyper reactivity, hypersensitivity might in fact be a pre-existing condition. Then we have those kids who we included in the data at age 14 who were the cannabis users. If you recall, there were 70 cannabis users, 70 controls. We had 38 of them come back into the scanner at age 19. So again, we have, this is the, kind of recapitulates the effect we saw at 14. So this is just the subset. Instead of 70, we have the 38 cannabis users, the 38 controls. We see again, the heightened amygdala reactivity at age 14. We show at age 19 that the controls actually show an increase in amygdala reactivity. This is true for these 38, but also if we look at the other hundreds of participants, this is the typical pattern. So this is sort of the typical neurodevelopmental pattern. And we see, however, that the cannabis using kids don't seem to show that accentuated response, that elevated response. Again, you can interpret this however you want. Is this some example of kind of precocious development? They're already at more adult levels and that's why they don't see a big change. Or is it because of their cannabis use in their intervening years? They were self-medicating for this, heightened amygdala response, and the 5 years of cannabis use have blunted their response. Who knows, right? One can pick the post hoc explanation that you like. But the point that I just want to make to kind of orient us into today's talk is, this is complicated. So we have, just looking at the amygdala, just looking at this task, we have heightened amygdala response characterized as uses at 14, but it also seems to predict use at age 19, and it also seems to change following escalating use between the ages of 14 and 19. So with this one probe, one sample, complicated. So disentangling all of this is the challenge, and that's what we're going to focus on today. So to give you a quick overview, so we just kind of cover that cannabis amygdala function. I will mention the enigma addiction just because you mentioned it, because I'm always looking for people to join the consortium. So I'm obligated to throw in a bit of a sales pitch in all talks. We'll talk a little bit then about causes. Can we predict cannabis use in adolescence? And then we'll talk about consequences. And there's two things I want to tell you about. One is a study we published a couple of years ago that I suspect none of you will believe. I'm not even 100% sure that we believe it, but I'm going to throw it out there anyway for your reactions. And then the second, this is actually unpublished. This is under review at the moment, and I think these are the most compelling data we have. These are 5-year longitudinal data in cannabis users that will speak to potential impact of cannabis on adolescent development. Okay. So let's get started. And I won't belabor this because this is not on adolescence per se, but just want to mention, just to reiterate what Michael said. So this is a consortium. We try to gather together, data sets that have already been, published and used, but use them for secondary analysis. The whole idea is to kind of build up a large sample size so we can do proper, reproducible analyses, and also to try to detect brain gene association. So clearly you need a large sample size. So if any of you are interested in joining, please, follow the website or shoot me an email. If you've got data that's just sitting in a can, this can give it a second life. We did, this might be useful for today's purposes, to kind of identify targets. So, we're going to talk about adolescents and their, relatively light levels of use. So, what do we know about adults who have cannabis use dependence? We did just published a couple of years ago an analysis looking at just cortical thickness across different substance dependent groups. So, pretty large sample size. And it's a relatively straightforward analysis, free surfer cortical thickness. What we found is, lower brain volumes, cortical thickness in individuals in substance use. These were, observed in a number of different brain regions. The most widespread effects were associated with alcohol use disorder. So across, alcohol, cannabis, nicotine, cocaine, methamphetamine, the biggest effects are seen for alcohol. The beauty of having all, these relatively large sample sizes, we can see, are there brain regions that are showing differences that are unique to some drugs, or that are actually common across drugs? So we can kind of disentangle drug-specific effects from general addiction effects. For today's purposes, when we compare the cannabis users alone against controls, we don't see differences. So whereas we do see robust effects in some of these other groups, for the cannabis users against controls, we don't see significant differences. And we can even throw, sort of sophisticated support vector machine classifiers. And the purpose of this is to see, okay, if we do a kind of multivariate analysis and try to capture any non-linear associations, is there any information here that would help discriminate cannabis user from control? And, we don't. So we can have, successful brain-based classifiers for, alcohol and nicotine and cocaine, but actually not for cannabis. We can, though, like I say, look at the addiction general effects. So there are just 7 regions that came through here in this analysis. So, this is just showing you the healthy controls, and then alcohol, nicotine, cocaine, methamphetamine, and cannabis. And you'll see for some of these effects, when we include the cannabis group in with the other substance using groups compared against controls, we do see significant differences. So on the left and the right insula, we see reduced cortical thickness. In the left and right middle temporal, probably not so much for cannabis compared to controls. So controls on one end and cannabis on the other. When we look at left inferior parietal, some evidence of effects, left supramarginal, and in right medial OFC. So, take home message from the adult cases and controls is that, there may be some associations between cortical thickness and cannabis use, but, none that seem to be unique or specific to cannabis use. Maybe, regions that are implicated in sort of more general addiction processes. Okay, but let's now get back to the teenager. So again, there's really 2 halves to this. So the first half, we're going to talk a little bit about causes. Can we, in cannabis naive controls, predict who's going to go on to use cannabis? And then the second half would be consequences, looking at low exposure effects and 5-year effects. So let's talk about causes. There's got to be at least one obligatory joke thrown in. So adolescence, as you know, is a period of rapid change, and I'll let you read the finish there. And those of you who have teenagers, and I have 2, might appreciate the sentiment there. I don't need to belabor this. I'm sure you are all very aware that not only are there substantial brain differences that happen during this period, but there's just very substantial physical, emotional, intellectual growth. Substantial percentage of psychiatric disorders emerge during adolescence. So adolescence seems to be an important period of time to understand, and that's our focus here. And this is just other data showing again that you do have these, rather substantial brain changes that occur during the adolescent period, reductions in frontal gray matter, increases in white matter. So this has, been fairly well characterized. The data set I want to tell you about, essentially you could think of it almost as a precursor to the ABCD study, which I'll mention very, very briefly at the end. This is a project called IMAGEN. It's a longitudinal study of teenage drug use. We have, neuroimaging of cognitive control, so a stop task or a motor inhibition task, reward processing, social processing, that was that angry faces task I showed you earlier, and of course, brain structure. It's a multi-site study. We also have, genetic data on substantial numbers of the kids. We have, fairly extensive psychological assessments, all the things you might imagine: drug use, personality, clinical symptoms, cognitive abilities. But the kicker is that, we're getting more used to having these large samples now, but this study started about a dozen years ago. So it was, I think, notably large for when it started. And the beauty of it is it's got about 2,000 kids, is that, we now have data spanning, 10 years in these kids. So they're all recruited for the age 14 assessment. So full imaging battery, genotyping, phenotyping, a quick follow-up at age 16. But then at age 19, we got about 1,500 of them to get back into the scanner. And at age 23, again, about 1,400 or so, I believe, to go back in the scanner. So we have, 10 years of longitudinal data. We've done very little with the age 23 data. We've just essentially completed the quality control on those data. So stay tuned, and there'll be more there. But what I'm going to focus on is, the age 14, age 16, and age 19 assessments. So, okay, so first, can we look at the data at age 14, all of it, and see, can we predict who's going to go on and be using cannabis 2 years later? So that's essentially that's the essence of the goal for this first analysis. So we identified subjects who were all cannabis naive at 14. Then we had about 173 who went on to use cannabis by age 16, and this is all, self-reported use. And then another 1,200 or so who remained cannabis naive. Again, we are not talking about a sample of kids with, cannabis use disorder. This is, you can see it there. Substantial numbers just report, between 6 and 9 uses, 10 to 19, and up to, 40 plus uses by age 16. One caveat, unsurprising, I'm sure you all know this very well. The future cannabis users, they also go on to use more alcohol, more nicotine, more illicit drugs. We tend not to try, in these analyses, we don't control for that, because I think you're, going on to these other drugs using cannabis, there's probably a lot of the same predictors for both. So you kind of, would be controlling away variables of interest. Instead, if we can identify the predictors of cannabis use, we might then do an analysis with another group of kids who don't go on to use cannabis, but go on to use alcohol, and see if they're the same or different predictors for the two groups. That's how we try to disentangle these kind of issues with polydrug use. This is what we include in the analysis. And I guess I'll give you all a moment to stop and think as to what you think might prove to be the best predictors of future cannabis use, right? So we have these kids some of whom go on to use cannabis, some who don't. And we're looking at the data at age 14 when they were naive to see, okay, is there anything in here that would predict future cannabis use? And the other thing I should mention is, we do this in using some machine learning methods with proper cross-validation. So these are, predictors that are shown to be generalizable to unseen data. But the measures we include, you'll see here, I know I'm not going through all the details of them because there's a lot to discuss, but we have a number of, psychometric measures. So, standard demographics, cognitive assessments, personality, stressful life events, whether at age 14 the kids had any smoking or alcohol, and also some parent measures, parent personality, parent substance use. We include a number of SNPs. It's complicated. We still don't have really a great grasp on how to accommodate the massive amounts of genetic information. So typically what we do for these analyses is we would identify SNPs of interest, based on published GWAS, or just that we think are inherently interesting. That's, dopamine related SNPs, things like that. And then a whole bunch of brain measures. So structural as well as functional region of interest measures. So essentially we've got 2,400 variables. We put them into this big machine learning soup and we see what comes out. Can we predict future cannabis use? And if so, what drives the prediction? Is it all of these? Is it some of these? Is it all in your genetics? Is it all in your brain? Is it all your parents' fault? These are the questions we're trying to address. How well do we do? First thing to note, I should mention this, is we did this separately for males and females because, there's might be good reason to think that the predictors for cannabis use in boys might be different than in girls. Yeah, and you'll see there at the bottom, this is the area under the curve. So these are receiver operating characteristic curves, so sensitivity against specificity. Two things I want to point out, this is how well the predictive models do on unseen data. So this kind of quantifies how well this predictive model generalizes. Now, this is the challenge when you got lots of subjects and lots of variables, you can very easily overfit to the data. So you want to make sure you got some sort of cross-validation scheme. But the other thing to note is that even though the model, was very significant for both, it actually did a better job predicting future female users than future male users. And what's curious about this is there are more male users than females, and the males use more than do the females. But nonetheless, we can predict females better. So, it's a curiosity. So there's something a bit more distinctive, perhaps, about the profile of future female user. Okay, what drives the prediction? Busy slide, but a couple of things to a kind of big picture first before we go through it. What we have here on the left-hand side are the predictors for the males. We have predictors for the females. You'll see some are common to males and females, some are unique. There aren't too many. Remember, we started off at 2,400. So the prediction is really carried by just, a dozen or so variables. And we've broken them down here by kind of, broadly speaking, the psychometric ones and the brain ones. So a lot of these, make sense. If you, at age 14, again, you're cannabis naive, but if your parent reports having used cannabis, then that helps predict that you will go on yourself and be a cannabis user 2 years later. And that's true for boys and it's true for girls. If at age 14 you've had any cigarettes, like literally just one, if you've had a cigarette or any alcohol, you're more likely 2 years later to, be reporting using cannabis. And again, true for boys and girls. If at age 14 you report any sexual experiences, distressful life events, and then some, personality variables, being higher novelty seeking and disorderliness, that's predictive. In the female model, there are some predictors that are unique to females. So being higher in extravagant personality, and then also some parent measures of personality. Whereas for the boys, scoring higher in deviant life events and other parental personality measures carry the prediction. The brain variables is where it gets interesting. It gets interesting and it gets complicated. You'll see that there are no brain variables in common between the boys and the girls. And we've explored this a bit in depth. If we take all these psychometric variables in the females, they do a pretty good job of also predicting cannabis use in the boys. And we have another group of kids who don't go on to use cannabis, but they go on to use alcohol. And, the variables that predict future cannabis use actually also tend to predict future alcohol use. And the variables that predict, future cannabis use in boys go on to also predict future alcohol use. But the brain variables don't show that pattern. So the brain variables that predict cannabis use in girls don't at all predict cannabis use in boys, and vice versa. And the brain variables that predict cannabis use don't at all predict alcohol use. So, be that whatever that might mean, it does suggest that there's interesting potential specificity in the brain predictors. Now, what regions are carrying the prediction? This is where it gets complicated. I mean, if you buy into these sort of, machine learning, exploratory analysis, you're going to have to live with the results that come through. So, there is no easy, I don't think, easily interpretable pattern. These are the regions that come through. Some of them are, gray matter volume reasons. Some are for the stop task. Some are for the reward task. Some are for the faces task. Some, if you want, you can make some, plausible post hoc stories. We are seeing some ventromedial prefrontal cortex, some orbitofrontal cortex as a predictor, some anterior cingulate. You wouldn't need to be too creative to figure out how they might be of relevance to addiction. But, we're also seeing substantial cerebellar regions, more posterior regions, temporal regions. So, interpreting what these variables might actually mean, are they inherently meaningful or not, is a challenge. So really the machine learning kind of gets us started, but doesn't answer a lot of the questions. But again, just the point I do want to make, the curiosity here is that, the brain variables were relatively weak predictors. So if you just want to do a prediction, personality and stressful life events will essentially carry all the prediction for you. That's all you really need to know. The brain variables are not as good predictors, but they do have this interesting potential to have greater sex and drug specificity. So this is something we want to follow up on. Okay, just trying to be cognizant of the time. So okay, moving on. So that's causes. Now I want to talk to you about consequences. And I think I have about 15 or so minutes. So I should be on the money here. So, okay, so two things I want to tell you about. The first is this low exposure effects that I'm not sure anybody believes, but I'm going to share it with you anyway. And you can tell me if you find it plausible or not. What we did, oh yeah, so I should just focus. I mean, maybe this is super duper obvious to all of you, but for the remainder here, we're not going to be talking about brain function, but just, brain structure. So, you probably know this, you can take your high resolution anatomical scans, you can skull strip, you can zero in on the gray matter mantle, you can measure the inner boundary, the outer boundary, and you can get, these nice kind of quantifications of cortical thickness measures. And if this looks like, good old fashioned phrenology, it is. This is modern day phrenology. Phrenology is a great idea. Just, the slight mistake was thinking that the skull told you something about the brain. But, there's a lot we can do in measuring the actual gray matter, the actual cortical thickness, even though it's an ambiguous measure for reasons we can get into later if you like. But this is what our focus is going to be for the remainder here. Okay, so here's a radical question. Can one or two uses of cannabis affect brain structure? What motivated this in part was some evidence from mouse models suggests that this is possible. So this was a study a while back showing that a single administration of THC could disrupt brain plasticity in a mouse model. So what we did again in the IMAGEN sample is we found 46 kids who reported that by age 14, by the time we scanned them, they had had one or two joints. They'd had one or two lifetime uses of cannabis. And then we went hunting and we found another 46 kids who were matched on all the things you see there. And again, just to reiterate, I mean, this is valuable. Being able to get carefully matched, controlled kids is, again, one of the, I think, the really nice strengths of having these large samples. So we get kids that are matched on age, sex, puberty, handedness, the site they come from, verbal performance IQ, SES, alcohol, nicotine, and indeed also on total gray matter volume. And essentially we plug these two in, and this is a super duper simplistic analysis. We're just looking at gray matter volume. Are there any differences between these kids? And we weren't really expecting to see much. And we saw a fair bit that kind of surprised us. And the effect here you'll see, and we can come back to this in a second, is actually greater gray matter volume in the cannabis using kids versus the controls in a whole bunch of these brain regions. And they're sort of shown in a little bit more detail here. I won't get, well, I'll just mention to you as a quick aside, we have seen other effects we published with that seem to suggest that even just, one or two cigarettes might change gray matter volume. But so, we were surprised, but we thought this is potentially plausible. Maybe could one or two uses of cannabis actually change your brain? So questions. You might think hardly. What's more likely to be the case is that there are just pre-existing differences that maybe predispose kids towards use, right? That this is not cannabis causing these brain changes, that if anything, maybe there are just brain differences that cause cannabis use. We don't think so. Again, I've just been belaboring, we've done these analyses trying to predict cannabis use, and the regions that predict cannabis use are different than the ones that I'm showing you across the top here. And if we look, just at these brain regions at the 14-year-olds and see if they predict cannabis use at age 16, they don't at all. So, we don't find evidence that these are pre-existing, that these up here are pre-existing differences. Next question, maybe these effects are driven by other phenotypic variables. Maybe this isn't a consequence of cannabis use, but maybe these kids are just, higher in sensation seeking, and that's what the brain difference is all about. Again, we don't think so. We looked across the full battery. The cannabis users, they do score higher in sensation seeking and in a measure of agoraphobia. But even if we include those as covariates, these gray matter effects persist. Is this at all plausible? One thing we did is, what we can do is we can look at the overlap of where we're seeing these effects and look at, see how that spatial map of where there are gray matter volume differences, does that correlate with maps of where CB1 receptors are? So you can, this is going to the Allen Brain resource, where we can look at CNR1 gene expression. So this is the gene that codes for the CB1 receptor. And you know, the plots might look a bit strange, but essentially we see a very, very strong association between where we are seeing these gray matter volume effects, differences, and where the CB1 receptor is expressed in the brain. So again, it adds a certain plausibility that these differences are related to the cannabinoid system, which again would be consistent with it, you know, being a consequence arising from cannabis exposure. The plausibility, just to kind of quickly touch on that, we're not sure, of course, what exactly is happening. One possibility for why this effect is greater in the cannabis use versus controls is that perhaps, like in that mouse model, what has happened is that we have a sort of, you know, delayed neurodevelopmental process. You know, for the most part, as I was showing you earlier, during this age period, you know, gray matter volume is reducing, cortical thickness, you know, you got synaptic pruning and so on and so forth. And there is actually what we think might be happening, but you know, we don't really know, is that maybe perhaps the use of cannabis is somehow disrupting that typical cortical thinning process. And that's why the cannabis using kids are showing up as having greater gray matter volumes. Are these effects consequential? So this is important. I mean, again, like I say, people get super duper hot under the collar about these effects. And, you know, we have to be clear, you know, it's kind of set it up here when we look for the phenotypic variables, there are very few differences between the cannabis using kids and the controls. So even if this effect is true, it may not be functionally consequential. The only thing that, you know, we can bring to bear here is if we look at, you know, some of the reasons where we're seeing effects like in the right and left temporal lobe, we see that greater volumes are related to lower IQ score. Our cannabis using kids are showing greater volumes, but there isn't a difference between our cannabis using kids and controls in IQ. However, maybe that's power. So if we had more participants, we might start to detect IQ differences, but we're not seeing it here. So functional, psychological, cognitive consequences, we're not really seeing it in these data. And then of course, the question that of course is near and dear to all our hearts is, you know, are these effects publishable? Well, you know, we did publish it, so the answer there is yes. But really, this is the sort of finding I think that you can get in human research that really, really, really would benefit from a proper experimental model. This is described as ongoing. That's actually not fair. This project actually never really fully got off the ground. So we're still kind of waiting to do this, to, you know, do a kind of parallel study in a rodent model where we have just one or two exposures and see what happens. But part of why I'm interested in this, you know, and I'm fully cognizant of the fact that this might all not be true, is that to my knowledge, this is very understudied. I mean, if you want to study, well, what are the effects of alcohol or nicotine or cannabis that's in the adolescent brain, it sort of makes sense that you'd start off with those kids who are using the most and compare them to kids who are not using. You know, it's going to maximize your effect. You know, is it possible that there might be brain changes that arise from first uses? Is it possible that that might prove to be consequential for then going on and having a second and a third use? We don't know. But to me, my understanding is that this is very understudied, both in human research and in animal models. So that's why we're kind of curious to pursue it a little bit more. Okay, the last example I want to give you is this study. So this, like I say, is unpublished findings. Again, same study, and this is the last study I'll talk. So apologies if I'm throwing too much data at you, but this is the last one. So here's what we did. So this, again, the IMAGEN study, we have brain data and drug use measures on these kids at 14 and again at 19. So we identified over 800 kids at age 19 who were all cannabis naive at age 14. So they're all naive at 14, we've got the baseline data, but who reported use by the age of 19. So they used between 14 and 19. And we simply tested if cortical thickness correlated with cannabis use. Again, these are the use levels. You know, the nice thing here is that the sample size is relatively large, but we have, you know, a substantial number of kids who didn't use at 14 and didn't use at 19. And then these kids, all of whom, none of them used at 14, but by age 19 had used once or twice, 3 to 5 times, 6 to 10 times, 40 plus. And we wanted to test to see, are there any differences in their brains that might be related in an almost like, you know, a dose response way to their cannabis use. So here's the result we see. So the linear mixed effects model, what the primary test of interest here is this interaction, age by cannabis, right? So we're expecting a main effect of age. There should be this kind of cortical thinning. The question is, does that age-related effect, is it, moderated by cannabis exposure? So the age by cannabis interaction is the critical test. And what you see is we see, fairly substantial effects, largely prefrontal, that show this interaction. We can unpack this a little bit. If we look just at the data at age 19, so this is just looking now again, cannabis use, does it correlate with age 19? Essentially, it recapitulates the previous interaction. So, that's the interaction map. And then this is the analysis at age 19. If we do the same analysis at age 14, we essentially see nothing. So what we're seeing here is an association with cannabis use that's at age, that's present at 19 and was not there at 14 prior to their cannabis use. So again, it suggests this was not a pre-existing difference. This is something that has emerged with their cannabis use over the intervening years. We can control for self-reported alcohol. Again, the nice thing about having a large sample is you can, stick in these covariates that are very often confounded. But even if we control for it, it doesn't make the effects go away. So we still see this cannabis, this age by cannabis interaction, even when controlling for alcohol. Just a couple more things to say about this. We can look to see, in the brain regions that are showing this cannabis effect, if I can call it that, overlap with regions that show the biggest age changes. So again, like I say, from 14 to 19, you're seeing a lot of cortical thinning. And a vertex by vertex analysis, those regions that are showing the biggest age-related changes also tend to be the ones that are showing the cannabis-related effect. So those regions that are most plastic, that are changing most, also tend to be the ones that show this cannabis-related thinning. We can do similar to what we did before. We can look at where in the brain are the cannabinoid receptors, and we find again a nice overlap. The brain regions where the cannabis receptors are to be found overlap with the brain regions that show this cannabis-related cortical thinning from 14 to 19. So again, it adds a certain plausibility that these effects are related to the cannabinoid system, which again would be consistent with it being a consequence of cannabis exposure. This slide just puts those two last bits and pieces together. So we have, and this is just a threshold map. We essentially have, brain regions that are showing the cannabis effect, correlate highly with brain regions that just show normal age-related change, and also correlate with brain regions that show that reflect where the cannabinoid receptors are, and then just puts them all together. Last thing I want to say about this, for those of you who are interested in these analytic approaches, there are ways, and these methods all come with different assumptions, that you can try to look at sort of the joint probabilities between these variables that are all correlated and confounded with one another, and develop putative causal models. This is a Bayesian causal modeling association. So we include a number of variables that you can see up here on screen. This is a polygenic risk score, age, sex, baseline measures of cortical thickness, tobacco use, alcohol use, and then at age 19. And you put these all into the analysis, and it tries to come up with the best model with kind of directional association, so causes that best fit the data. And very, very consistently, very robustly, we get this solution, where the best model to fit the data and all the patterns of joint of conditional probabilities is one where there is an association from cannabis use to a change in cortical thickness. So, some people like these methods, some people don't. But for what it's worth, it's another piece of converging evidence that this would seem to be the causal direction. Okay. Last point about this. Any cognitive mental health consequences of this? So, maybe, maybe not. The maybe is that cannabis use at age 19 was significantly associated with these measures of psychosis symptoms. But the problem is we didn't have that same measure, the CAPE, at age 14. So there is a possibility that these were pre-existing differences. So we just don't know, which is unfortunate. We do have measures of impulsivity. And what we find is that this accelerated thinning, if that's how we can interpret it, this age by cannabis effect in the right dorsal medial prefrontal cortex, it was related to attention and impulsivity at age 14. And in fact, we can do a mediation analysis. We can show that the change in lifetime cannabis use, the relationship between that change in cannabis use and change in attention impulsiveness is mediated through that thinning in the right dorsal medial prefrontal cortex. So again, it adds a certain plausibility to that these changes have some sort of psychological consequences. Okay, to try to wrap up. So what are we finding? So risk causes. Adolescent cannabis, I mean, it might seem obvious, but it's worthwhile saying it. Adolescent cannabis use, it can be predicted. And in fact, it can be predicted with a relatively small number of variables. Remember, we started off in that analysis with 2,400 and it does it. There's predictors, personality, life events, plus, as I've mentioned, brain measures that do seem to have this feature of being very drug and sex specific. This is the thing we're interested in doing. I have a T32 where the trainees work on these machine learning methodologies. So we're quite, curious to see, can we build brain-based classifiers that might help identify kids who are at risk for substance use and so on and so forth. The consequences, there's evidence, if you buy it, of brain changes arising from use, maybe even very light use in young adolescents. Of course, ongoing longitudinal studies, the IMAGEN study has a large sample, but again, it's just one study. So, we are following up here to see if they're in IMAGEN, now that we have these, new suite of data, a new wave of data at age 23, are any of those brain changes, are those impulsiveness changes from 14 to 19, what might they say about function and brain at age 23? So that's our ongoing analysis. And then, of course, because I spend most of my time thinking about ABCD, I'm obligated to include at least one slide on ABCD in every single talk. And just to let you know, I mean, you may know this, you may not, but this is ongoing. We are the 4th annual release is coming up. 12,000 kids. By design includes over about 2,100 mono and dizygotic twins. Annual assessments, biennial MRI, tons and tons and tons of data. I think if you take away all the neuroimaging and all the genotyping and all the hormone analysis and all the geocoding and everything else, there's still something like 60,000 variables per kid at each assessment. So it's a massive amount of data. And the point is that it's all publicly available. So if any of these questions are of interest to you, or, and this obviously ABCD goes well beyond just substance use into, a whole bunch of relevant issues for adolescent neurodevelopment. But, these are all publicly available, and we would strongly encourage you to, if you're at all interested in these matters, to, get access to the data and have at it. Okay, with that, I will stop and wait for Michael to field me some very soft questions. Thank you for your attention.
Great, thank you very much, Hugh. We do have a few questions. I often don't get to ask one, so I'm going to start one from myself, and then we'll go to Laura Holt and then Richie after that. So my question has to do with the single-use study, which on the surface does seem a little bit like one of those findings where you're not quite sure if it really will hold up. Do you think potency plays a role in that? So there's been a lot of change in the strength of THC in cannabis over the years. If you're using a very high potency THC strain of cannabis, a single use might have a much different impact than a lower potency or something that has a more balance between THC and CBD. What do you think about that?
Yeah, good question. Quick question. Should I stop sharing or keep the slides up, or what's the best way to work this?
You could stop sharing if you want. It's fine.
Yeah, okay. Great. Yeah, so the interesting thing about that, we don't really know, right? So this is a real challenge with these studies. You don't actually know what the kids are smoking. And part of that is because, the kids don't actually know what the kids are smoking. And we struggled with this even with the ABCD study. And there's no easy solution. And people once upon a time were saying, well, maybe we should just have kids bring, their joints to us and, give us one that we can then do an analysis. And you can imagine all sorts of ethical and legal ramifications of that. So we don't know. But the interesting thing in that particular study with the IMAGEN kids is that study is now 10, 11, 12 years old. So those age 14 data were collected quite a while back. So probably before this change in potency really kicked in. But It's a good question, and it's, like I say, it is it. And there's always this. I mean, this. I mean, these are crude measures. They're self-report. We don't have proper lab, validation of the measures. I do think, if you were to ask me, if this effect is true, we do know that cannabis sticks around in the system. Even 1 or 2 joints can stay in your system for quite a while. I suspect it's going to be very important the age at which the kids have these exposures, if it's true. So in effect that we might see peripubescent, I wouldn't necessarily expect to see, let's say, in 18 or 19-year-olds.
Great. So we'll go to Laura Holt next.
Thank you so much, Dr. Garavan. An especially fascinating talk, and to think about the potential effect of this single use. Just really interesting thing to chew on. My question, I think, actually relates a bit to Michael's question in terms of potency, but I'm curious just to hear some of your thoughts about vaping THC and how this is kind of factored into your, study protocols in terms of assessment, and how you think it might, could potentially affect the brain differently, or maybe even represent a subset of participants whose predictors might be a little bit different.
Yeah, I get really good questions, and I have nothing intelligent to say about it other than to fully agree. In the IMAGEN study, was even before vaping at age 14, so not an issue. But I know in the ABCD study, which actually, just as a small aside, I mean, the ABCD study, I think its origin was these very questions. There was kind of this interest to figure out what, given that cannabis is being decriminalized all over the country, what does it do to the developing adolescent brain? So that was the starting point for ABCD. But of course, now it got much, much bigger, and it's got to do with lots more than substance use. But all I can say is that in that study, there's a whole substance use working group, and they're very attentive to these issues. So the drug use and mode of drug use and vaping, et cetera, is assessed very, very regularly in the kids. And as new technologies and new drugs become more and more popular, then we kind of change the protocol to add those assessments. So I don't have the answer, but with the IMAGEN study, the kids are now 13, 14. So I would suspect in the next couple of years we would start to see substantial numbers of adolescent use, and then we can disentangle your question. But I can't at the moment.
Great, thank you.
Okay, we'll go to Rich next.
Hi, thank you for that presentation. It was really nice. So I have a question that I suspect you're going to have a lot of good points about why it's potentially a stupid question. So it might end up being more of a thought exercise more than anything else. Okay. So you'll indulge me here.
Yeah.
So what I'm thinking about is that, is particularly given that, at least in the studies that you presented, you're characterizing people who've never, and I understand that they're on the young side, but those who have never used versus who have reported like ever used, right? And what's likely is that the vast majority of those kids who have used, right, will never really develop any sort of substance use problem. Even if they engage in kind of like problematic use later on in adolescence, right, a good chunk of those kids will actually age out of that, right? Yeah. And so I wonder if, the way it's the presentation that's framed as if these are differences that we see as a result of use, right? I wonder if there's any benefit to think about it as that in some respects, because that's normative, that what you're seeing is also the results of those kids who don't. So in other words, it's not that the changes are in the kids who use, at least in terms of how it's conceptualized in the study, but the kids who have never used.
Yeah, so a couple of things. It's an interesting one. I think when we do these analyses, sometimes my colleagues query, maybe this is where you're coming at too, the controls, these super clean controls who never use it, are they the appropriate comparison group? What you might want to actually, what might be more interesting is kids who've tried cannabis, but were, not so much, not for me. That might be a more interesting sample to have, because one might argue with maybe not so much with cannabis, unless you're here in Vermont, where essentially it's it's everywhere. But alcohol, where it's essentially normative, maybe the kids who are super clean are a different cohort or have other sorts of, environmental strictures, et cetera, that, and also then, maybe they have latent risk potential, but it's just never been expressed because they've never used. So big picture answer to your question, because I like it as a kind of thinking aloud, is, yeah, I think comparing to kids who've tried but who don't develop any kind of problematic use levels, I think is a very sensible analysis approach. And again, hopefully in a study like ABCD, where we have a much larger sample, and then we have much more frequent assessments, like we have assessments on these kids every 6 months, we will have much better characterization. And, the IMAGEN study, 14, 16, 19, it's spotty. It doesn't have that kind of fine grain that you're talking about. The other thing I would say, and it's kind of reason why I mentioned the enigma at the beginning, one of the things we're interested in doing is, so these kids who, let's say it's all true, and you see these volume reductions as a function, but whether or not that's consequential, like, we don't know. Like, time will tell. So clearly we need these longitudinal studies to go for decades. The fact that we now have age 23 data will be very interesting in the IMAGEN sample, because we might have some people who are developing substance use dependence problems, and then we can go back and look at predictors specifically for them. But one of the things we can do at the moment is we work with the enigma data set. So here we have, like, bona fide cases, people with dependence versus controls. And we might, like, create a brain-based classifier on them. And then we take that and now we apply it to these longitudinal cohorts, to these kids. And we see, okay, does this brain signature, this classifier of dependence per se, does it characterize problematic users? And in some cases, we've seen that. So again, I won't bore you with the details, but we have some sort of measures of, structural covariance, correlated patterns of cortical thickness that we see to be present in alcoholics. And we saw it in IMAGEN in the problematic, not alcoholics, but, high audit scores. We see the same effect. And then in those kids, in some of them, we went back and we looked at their data at age 14 before they had any alcohol use, and they showed the same effect. So, I'm just kind of, riffing on what you're saying, that I think this is one kind of approach that we can get at it. We look at bona fide users' dependence and then use that to kind of then get a better understanding of what's normal variation, what's potentially problematic brain signatures in these adolescent studies.
So we have time for one more question, and then I have one general question about the ABCD project that we'll close with. But let's go to Laura Martin. Her question actually seems kind of related to what you just talked about, so it might be a little bit redundant, but Laura, go ahead and ask your question.
Yeah, I do think you've pretty much already answered this. I was just wondering if you've had a chance to look at this, or this may actually be a question for the ABCD trial, whether there's slowing or reversal of this clinical thickness change if someone used and then quit using or changed their use patterns, slowed it. Do you see the associated changes?
Yeah, that's exactly the sort of question we want to ask with ABCD. In IMAGEN, we just don't have enough kids to fit that profile. But it's, this is one of these questions that, it makes you realize just how little we understand. Like, we've no idea if there are these early effects, whether or not they go away if you stop using, or are there critical periods where it might have long-lasting effects. Again, you can look at some of the animal models. I was just looking at a paper there out of Yasmin Hurd's group, where they are showing that some effects in kind of pyramidal cells and prefrontal prelimbic cortex in a rat model, that and then you have good, you can control the cessation of exposure, and then into adulthood they still see effects. So, but what's happening in the humans to see those different profiles of use? Yeah, we need lots more data. So hopefully the ABCD study will answer it.
Great.
Well,
thank you.
On that note, I wondered if you could spend just a minute or so. We have a lot of trainees on the call and a lot of faculty members that might be interested in using ABCD. So could you give sort of the 10,000-foot view of how one gets access to that data and sort of how it works? Yeah.
Well, the first thing is, stay well away from being involved in collecting it, because it'll do your head in. No, so it's all up on the NIMH data archive. So you go to ENDA, the NIMH data archive, and there's a permissions process. And all you have to do is there's a data use agreement. And, it's unsurprising. You just have to promise that you won't try to identify the participants because, there's such rich data there. They are potentially identifiable. And you have to promise that you won't reshare the data. And then you have to get an institutional signature on it so that it has a little bit of teeth. So somebody in your grant's office will, sign off on that. And that's it. And then you download and you do whatever you want. There's no, there's no process for, where you have to make a proposal or you have to worry if you're competing with somebody else's proposal. You just have full unfettered access to the data.
So you don't need unique IRB approval at each individual institution as long as you have that institutional signature.
Yep.
Okay.
And there's also, they put a lot of time into creating a data analysis portal. They call it the DEEP, Data Exploration Analysis Portal, which is really, really nice because after a while, downloading the data is going to become very, very challenging because, there's 12,000 kids, brain image, very high res brain imaging. So the data analysis portal, the nice thing about it is it has, very nice interface. It's got data dictionaries. You can kind of search for a term, you put in weight and all the variables that might have something relevant to do with weight will come back at you. You can select them, you can see distributions. It will access the most up-to-date quality control version of the data. It'll run your analyses. I'm sorry, I'm kind of rattling on here a little bit, but the statistical models are very complicated because you've got siblings and twins, so they're that are nested within a site, within a scanner, within a site. So you got these nested random effects models, and the DEEP controls for all of that. It has all the appropriate statistics built in and all the appropriate covariates. And so the beauty is that then you can work with the data just through this interface, meaning you don't have to get into the business of downloading, what will become, petabytes of data in years to come.
That's good to know and very helpful. So on behalf of the Cofrin Logan Center leadership team, I want to thank you, Hugh, for taking the time from your busy schedule with all of the many things you're doing to share the talk today. We're going to put the link to our website and Twitter in the chat one more time for those of you that are interested in our upcoming brown bag talks and seminar talks. Just to give you a preview, we'll be closing out our spring 2021 seminar series with a talk by Dr. Katie Wickowitz from the University of New Mexico. We'll be coming to talk about recovery and other issues surrounding how we define recovery from alcohol use disorder and some of her recent work. So thank you very much, Hugh, and thank you for everybody on the call today.
Thank you. Take care.